Hi everybody, today I'm going to do a video on the big idea sheet, gene technology. So these are a lot of the labs that we did, like gel electrophoresis and colony transformation. So we're going to be going through those. I have the words already written on the page, so if I start going too fast, just pause me. First question, what's the purpose of forming recombinant DNA? And so the purpose is to create an organism with an additional gene, something beneficial, something advantageous. So a great example is agriculture. We have been able to manipulate crops so that they can have extra genes, that maybe they have extra nutrients because of their ability to transcribe and translate that DNA, or maybe they are insect resistant or they are pest resistant in some way, so things aren't eating them. Maybe they are cold resistant or drought resistant, something to give them a better chance of survival. A gene that another organism had is cut and taken out of that organism and put into this organism. That's what recombinant means. Another great example that I wrote here is bacteria has been altered so that it can contain the human gene for insulin. And bacteria produced insulin until like 10 years ago. Now we synthesize it, but all of the insulin that diabetic patients needed used to be synthesized by bacteria because we gave them our gene. All right, next question. How can genes within a chromosome be isolated? So that means, like, how do you get it out? How would you isolate one gene if there's 1,000 different genes on a chromosome? And you use restriction enzymes. Restriction enzymes are very specific. They only cut at palindromes, and there are many different types of restriction enzymes. You guys used ECHO R1, HIND3, BAM. Those are some examples of restriction enzymes. All right, next question. Draw out the steps of forming recombinant DNA. So you might want to do this as a picture. I just actually wrote it out as words. The first step is you have to treat them both with the same restriction enzymes. So when you're treating the DNA, you're cutting it, and if you cut them with the same restriction enzymes, they're going to have the same sticky ends. So you're going to have a spot that you're going to be able to add the new piece of DNA to in the existing organism. The second step is you have to get them to take in the plasmid. So it is a heat shock process. You put it on ice and then warm and then back on ice, and it alters the permeability of the membrane and makes it more likely to take in the plasmid. So hopefully you are remembering that we did this with E. coli, bacteria. We grew it and then we treated it with a plasmid that had two genes. There was a gene for ampicillin resistance and there was a gene for fluorescent protein so that it would glow. And the last step is just allowing it to express. So if you give it time, it should express those genes, therefore go through transcription and translation and build the proteins. And the proteins are evidence that it has actually taken in the gene. All right, next step. Describe how PCR is used. PCR is just to make a lot of copies of a gene. So if I've isolated something like the breast cancer gene, I want to make many, many copies of it before I do any research so that I always have backup copies. And other labs can also use that gene and do copies. So that's what a PCR machine does. All right, next question. What is gel electrophoresis used for? So a gel is used to separate fragments of DNA. It's based on their size and based on their charge. So use the following diagram to explain how gel electrophoresis works and how the results are analyzed. So I have this diagram here and it shows quite a few things. So right here, these are the wells. This is where you load the DNA, and it has to be pre-cut with the restriction enzymes before you load it. Usually in the very first well, you want to load a standard. A standard, you know the piece of DNA that you put in there, and you know how many times it's been cut. So you know how many bands you'll get, and you know the size of each fragment. You know the size of the fragment in base pairs. So for example, if I know that it has 3,500 base pairs, I'm going to be able to compare other bands that are run on the same gel at the same time, and if they're around the same distance that they migrated, they're around the same size. All right. When you look at the banding pattern that results, you notice quite a few things. So I circled over here. This last well shows me that it was cut twice with a restriction enzyme, resulting in 3 fragments. The first fragment, the largest fragment, is up here. The second fragment is right here, and the smallest fragment traveled the furthest. This is the negative electrode, and the other end is the positive electrode. DNA is negative, so it moves from the negative end to the positive end. I also marked here that when you notice that a band is thick, that just means that there's more of it present. Sometimes you get really thick ones up by the well, and that means the restriction enzyme didn't work and it didn't cut your DNA. All right, so this last part, DNA is negative, moves to the positive end when you turn the current on. You'll remember that when you hooked up this machine, you had it in a buffer solution, and you had to turn on the power so that the electricity allows the fragments to move through the gel. All right, next question. This is a graph with dots indicating the points plotted of the standard. Draw in a standard line, it's not a curve, it's a line, and explain how this was formed and use it to determine the size of a fragment that moved 8 millimeters. So let me just remove the screen so that I can show you this at once. So the first thing I did is I made a best fit line and I hit as many of the points as I could. Then that standard, I knew the number of pieces that I had and I knew their sizes. And that's a comparison for any other fragments that are run on the same gel at the same time. So I can now take, like for example, I knew that this first point, that was 50,000. And I went and I measured on the gel how far it moved, and it moved 2 millimeters. So I went to the 2 millimeters, I marked that it was 50,000, that's how I got the point on the graph. This next one, it moved 6 millimeters, and I knew it was 10,000 base pairs. So that's how I got the points. And then I drew the best fit line. And then my task was, if it was 8 millimeters that a fragment moved, how many base pairs would that be? So you go to the 8, you go up until it crosses the line, and you go over, and it looks like it would be about 9,000 base pairs. Again, you doing it on your paper might come out a little bit different, but you get the idea. All right, what are RFLPs used for? So an RFLP is a restriction fragment length polymorphism. So a polymorphism is the fact that there can be many shapes or many sizes. A restriction fragment is a piece of DNA, and the length is the size of it. So there are many different lengths that you could find if you cut DNA with a restriction enzyme. If you tested my DNA and someone else in the class's DNA who was also a girl and also white and also, had this, the more things we have in common, chances are our DNA is going to be very, very similar. There's very few differences between human DNA. We are the same species. So you have to look for the introns, and you have to look for areas where you could be different in your DNA sequence. Those differences exist in what are called RFLPs. So if they were running a gel with crime scene and then multiple suspects, they are going to notice the difference only if they run specific introns. If they tested you all for your DNA for hemoglobin, it would come out identical for everybody. So that's why they have to use RFLPs. All right, next one, number 9. Describe how to clone. So Dolly the sheep, how did they get it? So the first thing they had to do was take the nucleus out of the surrogate mother's egg cell. So the sheep that was going to be the mom, they took the nucleus out of her egg cell, but then kept the egg cell itself. Then they took a diploid nucleus out of the donor. So this other sheep that was donating the DNA took a normal body cell, took out that diploid nucleus, and then put it into the surrogate mom's egg cell. So it never went through fertilization, but it actually was 2N. Then implant that egg cell back into mom and have it develop into a growing embryo. That certainly makes it sound a lot easier than it actually is. But if the pregnancy takes, meaning the embryo embeds itself in the uterus and it grows, then eventually that sheep is going to be born. When Dolly was born, Dolly had a black face because the donor sheep had a black face, and it was the genes from the donor. The mom, the surrogate mom that gave birth to the baby, had a white face. And they did that so that as soon as the baby sheep was born, they would know if it was the right clone and if it worked or not. So it was proof that it worked. The baby's going to also be identical to the donor, not to the surrogate mother, obviously, with the black face versus the white face. All right. When we use bacteria to clone a human protein, we insert a gene with no introns. How is this possible, and what do we call the DNA that's lacking the introns? So what we do is a process called reverse transcription. So if you start with the mRNA, the introns have already been taken out. So if you take the mRNA and then put it back to DNA, that's reverse transcription, and it uses an enzyme called reverse transcriptase. If there's no introns in that new section of DNA that was made from the RNA, it's called cDNA. The C stands for complementary DNA, but I like to think of it as perfect DNA, meaning it doesn't have the introns, so the new organism doesn't have to try and figure out which parts are nonsense and which parts are important. All right, next question. On to the next page. Oh, okay, so that was the end of the gene technology. So just as a refresher, make sure that you look through what we did for our colony transformation lab and what we did for our gel electrophoresis lab. Those were the key points of that big idea sheet. All right, this next big idea sheet is about mitosis, meiosis, reproduction, and variety. So let's start off here. What's the purpose of cell division? So when a cell goes through mitosis, it's just dividing and making more cells, and those cells are identical. Those are for growth and repair. Next question. Describe the organization of the chromosome during cell division. Include the terms centromere, chromatin, and sister chromatids. So when a cell is just an interphase, you can describe the chromosomes as chromatin. I like to think of the N at the end of it as like network. So a network of DNA looks all stringy like a net, and you can't really see individual chromosomes. When it goes through prophase, the chromosomes condense and thicken and become very visible. They have also duplicated during S of interphase, so you can actually see an X. The X is two sister chromatids attached to each other with a centromere. On the outside of the centromere is a region called a kinetochore, and that is where spindle fibers attach to. Number 3. A chicken has 78 chromosomes in its somatic cells. Those are the body cells. How many chromosomes did the chicken inherit from each parent? 39. That's okay, I'm recording. So how many chromosomes will be in each somatic cell after mitosis? So once it has divided, it will have 78 chromosomes, just like the original cell. How many chromosomes are in the gametes? 39, half. So a cell, when it is diploid, is 2N, 2 of each chromosome, and when it is a gamete, it only has 1 of each chromosome, so it is just N. All right, each of the offspring are going to have 78 because fertilization restores the diploid number. All right, briefly describe what's happening in each phase of the cell cycle. So a cell cycle is made up of interphase and mitosis. Interphase has 3 steps: G1, which is growth; S, which is replication of DNA, the S stands for synthesis, and G2, which is another stage of growth where it's really important to replicate all of the organelles within the cell, because once it goes through division, it's going to have to divide those organelles between the 2 cells. Then mitosis is the dividing of the nucleus. That is followed by a stage called cytokinesis, which divides the cytoplasm and makes the cells fully independent of each other. The stages of mitosis are prophase, metaphase, Anaphase, and Telophase. Those 4 stages are what allow the chromosomes to line up at the equator and then be pulled to opposite poles. All right, what regulates the timing of the cell cycle? So there are cyclins and cyclin-dependent kinases. Both of those control by their level what stage is going on. There are checkpoints. There is a checkpoint between G1 and S. There is a checkpoint between G2 and M, and there's a third checkpoint at anaphase. Those 3 checkpoints all allow a cell to go through the normal timing and allow it to have sort of a checks and balances to make sure that it's appropriate to move on. So the first checkpoint is based on cyclin levels. As cyclin levels go up, it shows that a cell is preparing to be able to go through mitosis. It will not go through S and replicate its DNA. All of the chromosomes will become sister chromatids, copies of each other's, only if cyclin levels are high enough. For the next checkpoint, the cyclins attach to cyclin-dependent kinases, so special proteins that are within the cell. When they attach to each other, they're called MPF. Maturation, promoting. Maturation promoting factor. A lot of people think of it as mitosis promoting factor, but it actually stands for maturation. Those MPFs allow a cell to move into mitosis, and then it will be able to divide. If there's not enough MPFs, then it won't move past the checkpoint, and it will not begin mitosis. All right, the last checkpoint, I don't have a spot for it on here, but the last checkpoint is during anaphase. And before the chromatids are pulled to the poles, there is a checkpoint where the spindle fibers yank a little. And if there's tension, then they know that the spindle fiber has attached to the kinetochore. If they've attached to the kinetochore, then they follow through that checkpoint and go through anaphase and split the chromosomes to the opposite poles. All right, next one, advantages and disadvantages of asexual and sexual reproduction. So asexual is good because it's very fast and the organisms do not have to be very mature to go through it. Bacteria can go through mitosis in 20 minutes, whereas sexual reproduction requires a lot of maturity, usually longer amounts of time, more of an investment, but it allows for variation, and variation is important for the survival of an overall species and a population in one area. If there's variety in a population in an area, a virus can come in and it's not going to wipe out everybody. If things are asexually reproducing and a virus comes in, their DNA is all the same, so if it kills one, it's going to kill all of them. Resistance is usually not in the normal chromosome, it's in a plasmid chromosome, so that's the only way that you have some of those bacteria that survive an antibiotic is if they happen to be resistant to it. All right, next one, define a somatic cell. That's just a body cell. A gamete, that is a haploid cell that is either a sperm or an egg. Homologous chromosomes, those are the chromosomes that are pairs. So we have 23 sets of homologous chromosomes. So we have 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, and so on. The one original came from mom in the egg, and the other original came from dad in the sperm. And sex chromosomes are the final pair of chromosomes, chromosome 23. It could be 2 X's forming a girl, or it could be an X and a Y forming a boy. And an autosome are the chromosomes 1 through 22 that code for all regular traits, not your sex. All right, how are traits from parents transmitted to their offspring? So every gamete has 1 of each chromosome. Every chromosome has thousands of genes on it. All of those are transferred by the process of meiosis into an egg or into a sperm. Now you get this unique combination because an egg only has half the chromosomes from mom in it. So you get this unique bunch of 23 chromosomes. There's going to be 1 of chromosome 1, 1 of chromosome 2, 1 of chromosome 3, but there were 2 choices of each chromosome. So that's why you and your siblings don't look identical. You each were a unique egg and a unique sperm from your mom and dad coming together to give you that unique combination. All right, how does the alternation of meiosis and fertilization maintain the normal chromosome number? So meiosis starts with a diploid cell, and the diploid cell goes through meiosis and produces a gamete, and the gamete only has N, half the chromosome number. Then you put two gametes together at fertilization, and that restores the diploid number. So it goes back to 2N for the zygote, and then the zygote goes through cleavage and divides until you have an embryo. All right, last page here. All right, what are the differences between mitosis and meiosis? And I really recommend for stuff like this, obviously it's right here on the screen, but go through this yourself. See if you can remember each of these. It's a great way to study. I'm not going to go through it right there because I feel like I've said everything that is on that chart already. All right, next one. Give a description of how meiosis 1 and 2 reduce the chromosome number. So, during meiosis 1, this is really important because you actually end up with haploid cells after meiosis 1. So it's called synapsis, and it is the pairing of homologous chromosomes. And they are copies of each other, so they look like two X's coming together. So chromosome 1 replicated and chromosome 1 replicated come together and form a tetrad. The way they line up is independent assortment. That means that chromosome pair 1 might line up mom on this side, dad on this side. Chromosome 2 might come in with dad on this side, mom on this side. And when I say dad and mom, I mean the chromosome that originally came from mom in the egg and the chromosome that originally came from dad in the sperm. So there's a unique lining up, and during meiosis 1, the pairs separate from each other. They still are X's, but now the cells are haploid because there's only one of each type of chromosome in each cell. It's still replicated, but it's still only one of each type of chromosome. Meiosis 2 then separates the sister chromatids from each other, resulting in 4 cells, and each of them are haploid. All right, discuss the ways in which genetic diversity is created through sexual reproduction, include independent assortment, crossing over, and random fertilization. So I will have those show as I'm talking about each one. So independent assortment is what I just spoke of, where each pair of chromosomes independently lines up at the equator. So you have all of these different combinations. So if you think of some possibilities, you've got 23 pairs of chromosomes. Maybe it's going to line up maternal, 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 paternal, maternal, 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 paternal. That is just a random grouping. Maybe the next time the chromosomes line up, there's going to be paternal, paternal, maternal, maternal. It's any random way. And because it's any random way, the offspring that result are going to be different from the parents, okay? Each egg is not identical to mom. It's only half of mom's chromosomes. All right, the next one, crossing over. That is when, during synapsis, the chromosomes are really close to each other, forming that tetrad. They can actually break off and switch homologous genes. If they switch genes, it's the only way that you can separate linked genes. So things like red hair and freckles, we expect them to be transmitted together. They're on the same chromosome. But sometimes there's people with red hair that have no freckles, and that's due to crossing over. The separation of genes happened because a gene broke off and separated. The further apart they are on a chromosome, the more likely it is. The closer they are together, of course, it's less likely for them to separate, so there's less of a chance of that crossing over. All right, random fertilization means you never know which sperm is going to make it to the egg to fertilize it. You also don't know which egg is going to be released that month. So random fertilization means there's all these possibilities of sperm and egg, and the ones that actually come together, that is random. Since they're each unique, you get these unique possibilities. All right, number 16. Fruit flies have a diploid number of 8. Honeybees have a diploid number of 32. Assuming no crossing over, is the genetic variety among offspring from any two parents likely to be greater in fruit flies or honeybees? Well, honeybees, of course, they have more chromosomes, so there's more possibilities. There's more genes, and therefore it is just more combination choices. All right, number 17, yours is missing this diagram. So Sorderia is a fungus, and the fungus, I drew a picture of it here. They have fruiting bodies called asci. They're an ascospore. So each asci holds 8 of these ascospores. So you can think of it like when you step on a fungus, like it releases all the spores up into the air. Those spores are actually in these cases called asci. There are black and tan ascospores. You could either have all black or all tan, or and that would show, like, let's say black is due to the capital B gene. If you were big B, you would see all black ones. If it was little b, you would see all tan ones. If it was big B, you're going to see 4 of one color and then 4 of another color. But when there's crossing over, you actually get a switch in the middle. So if you get 2 of one color and then 4 and then 2 of the other color, that is a perfect example of crossing over. It has broken on that part and switched. So to figure out the chances of crossing over, you look through all of these. So like, look at this one, 4 clear, or 4 tan, 4 black. But then this one, 2 tan, 4 black, 2 tan. That is the result of crossing over. So the ones that are the result of crossing over, I put check marks next to. Then I counted them up. 4 out of 12 had crossing over. That means the chance of crossing over is 33%. That means those genes are not very close to each other on the chromosome. All right, this last one I would like you to do by yourself first. It is a much trickier one. So go ahead and solve that. And when you're ready, stop, pause, and play again. And you can look at what I got. Here's the answer. As I went through, I did the L to S first because it was 20 apart. But then I quickly realized that there was going to be something going past either the L or the S because the numbers just weren't working out right. So I figured out that 5 and 7 add up to 12. So if I put A, then R, then S, it actually worked out with the 5, the 7, and the 12. It also meant that 8 was left between A and L, and I knew A to B was 18, so it showed me that the B had to be on the other side of the L for BL to be 10. So that is how I calculated it. All right, I hope you've learned a lot. Go back, study, review, and use your review notebook.